So welcome guys, welcome in another video tutorial. So today we will going to discuss about an interesting thing known as a-elation or attaching and effacing elation, or it is often represented as a/elation. So let's talk about this. So first of all, I want to told you that it is caused by EPEC. So I will first talk about EPEC, then I will move on that A by elation. So EPEC causes acute and chronic pediatric diarrhea, okay, especially in children of less than 1 year of age. Okay, this is important thing. So you all know, I think, that there are EPEC, EHEC, ETEC, EAEC, these 4 type of different strain available which are responsible for pathogenicity in E. coli, right? And almost all of them causes some kind of diarrhea. ETC causes watery diarrhea, EHEC causes bloody diarrhea, EPEC, here we have learnt about pediatric diarrhea, so there is a chance that we can forget. So the interesting mnemonic is, here we can see P, and here also P. That means EPEC, that means the E. coli strain which consists of a P will cause pediatric diarrhea. So that is the idea to remember. So EPEC is especially occurring in children under 1 year of age. So how they do this? They do this by causing dissolution of the microvilli of the enterocyte. So what is microvilli? Microvilli or microvillus are nothing but microscopic cell membrane protrusion, okay, that causes increase of surface area and helping diffusion, okay, and also it minimizes any increase in the volume. By doing this, they participate in 4 different functions. One is absorption, second one is secretion, third one is cellular adhesion, and the last one is mechanotransduction, okay. And as EPEC causes dissolution of this microvilli, so it is obvious that these 4 type of function will be hampered, right? So that is how they facilitates pediatric diarrhea, and also they facilitates initial attachment of the bacteria to enterocyte, okay. But the main hallmark of EPEC infection is this A by elation, which is usually occur in the intestinal epithelium. So A by elation, the full form of A by elation is attaching and effacing lesion. So it is the hallmark of EPEC infection, and it is occurring in the intestinal epithelium cell. So it is mainly caused by genes located in the pathogenicity island locus of enterocyte effacement, okay. So now the question is, what is pathogenicity island? So whenever we are talking about any kind of island types, first thing that is came into my mind is the Andaman Nicobar. So is there any kind of Andaman Nicobar types thing in present inside the bacteria? No, that is not. But the meaning of pathogenicity island is nothing but the gene that it acquires via horizontal transmission. That may be conjugation, transformation, transduction via any kind of transposon or anything, okay. That means pathogenicity island is nothing but a foreign gene that bacteria acquires, and this pathogenicity island helping bacteria to cause that particular infection. So here the name of this pathogenicity island is locus of enterocyte effacement, that is LEE. So the genes present in this locus of enterocyte effacement encodes 3 different type of important things. One is the adhesin intimin, which is responsible for adhesion. Second one is 6 effectors, including one essentially translocated intimin receptor, and the another one is a type 3 secretion system, that is T3SS. Okay, so these three things, along with there are 17 additional effectors present outside that pathogenicity island. Okay, they may be present on insertional element and prophages. So these 4 different things facilitates the attaching and effacing lesion in the intestinal epithelium. One is adhesion intimin, second one is 6 effector including one TIR, another one is a type 3 secretion system, and another one is 17 different effectors that is encoded by genes located on the insertional element and prophages. And we have already discussed about TIR, where we have talked about the pathogenicity of EHEC. So we know that TIR and intimin can bind with each other and facilitate the attachment of the bacteria to cells, right? So that is how they facilitates this attaching and effacing lesions. So that is why the name attaching, because here attachment is going on. Obviously we can easily understand that as adhesin intimin is present, and among 6 effectors there is one translocated intimin receptor. That is why it is obvious that attaching will occur along with effacing lesion. Why? Because here the genes which are responsible for A by E lesion, it is coming from locus of enterocyte effacement. Okay, so that is why the name A by E lesion, and it is causing by again 4 different type of things that is encoded by mainly 2 type of gene. So one is genes which are located in pathogenicity island LEE. So there are 3 things that is encoded by genes which are located on this pathogenicity island. One is our adhesin intimin, second one is the 6 effector molecule including the TIR and the type 3 secretion system, and here 17 different effectors are coming from gene located on the insertional element along with prophages. So that is how A by E lesion is occurring. So we can diagnose EPEC by demonstrating the localized adherence to Hep2 cell in cell culture and by agglutination test. So that is what we have discussed today. So EPEC is the E. coli pathogenic strain that causes pediatric diarrhea, that may be acute or chronic, and especially occurring in children under 1 year of age. The second one is it causing this by facilitating dissolution of the microvilli. Okay, and microvilli is what we have discussed, and also it facilitates initial attachment of the bacteria to enterocyte. And the hallmark of this EPEC infection is A by E lesion, that is attaching and effacing lesion. So attaching and effacing lesion is causing by genes located on the LEE, which is locus of enterocyte effacement. So this is the genes located on this pathogenicity island producing 3 different type of protein. One is adhesin intimin, another one is 6 different effectors including one translocated intimin receptor, and the third one is a type 3 secretion system, that is T3SS. Along with that 17 effector proteins extra are coming from genes which are located outside LEE, especially on insertion element and prophages. Okay, and we have also discussed the diagnosis, how we can diagnose them. We can diagnose them by demonstrating the localized adherence to Hep2 cells in cell culture and with the help of agglutination test. So that is for today, friends. So today we have discussed about the attaching and effacing lesions. So thank you for listening to this class.